Hello students, this is Professor Bridget again. We are now entering the final chapter of our study, chapter 27, the reproductive system. This chapter contains both the information on the female reproductive system and the male reproductive system. I'll start with the male reproductive system. But before we do that, let's just kind of understand some of the terminology first and the makeup of both the female reproductive system and the male reproductive system. So the components of both would be what we call the primary sex organs, or the gonads. In males, the gonads are the testes, and that's where sperm are made. And in females, the gonads are the ovaries, and that's where ova, or the eggs, are made. So both sperm and ova can be collectively termed gametes. So those gonads produce these cells called gametes. Both male and reproductive systems include accessory structures, just like we've seen in many of the other systems. Those accessory structures are going to include ducts through which the sperm and or ova travel, glands that are going to secrete some sort of product that assists in getting sperm together with egg. And then also, both male and female will have external genitalia. There's another component of both reproductive systems as well. It's more of a physiological component, and these are the sex hormones. In males, it's what we call androgens, the primary androgen being testosterone. There are a couple of different versions of testosterone that we all refer to as just androgens. And in females, the primary sex hormones are estrogen and progesterone. So those sex hormones come from, in both males and females, the gonads, so the ovaries or the testes, and or the pituitary gland. So those sex hormones play a variety of roles. They can assist with the development and function of the reproductive organs. So the hormone testosterone, for instance, has many functions, but it assists with sperm maturation or sperm development. Estrogen in females has, again, another variety of roles, but estrogen, just as an example, will play a role in building the inner lining of the uterus in preparation for a fertilized egg to implant. Sex hormones also play a role in growth and development of other organs and tissues. So think back throughout the semester to times when we discussed the effect of testosterone on a particular organ or tissue, or the effect of estrogen on an organ or tissue. Let's take estrogen first. Way back in Unit 1, we learned about osteogenesis, or how bones are formed. And we learned about the condition called osteopenia and osteoporosis. Remember that as women age, estrogen decreases, and that causes a decrease in the activity of osteoblasts. And remember, those osteoblasts secrete ground substance and fibers. So in this case, estrogen plays a role in the development of, or growth of organs, in this case, bone. Tissue, for example, here's a tissue one. Testosterone plays a role in muscle development. And then lastly, hormones or sex hormones play a role in secondary sex characteristics. So that could be things like armpit or axillary hair, hair in the groin, what else? Hair on the face, the development of breasts in the case of estrogen, and a number of other secondary sex characteristics. A secondary sex characteristic is a characteristic that makes you secondarily male or female. So it means a characteristic or something that you have other than male internal and external genitalia or female internal and external genitalia. These would be alternative characteristics that would most often be associated with a specific sex. Now remember, there are variations amongst individuals here, and not everybody has exactly the same secondary sex characteristics, and they may exist on a continuum as well. So moving on to the male reproductive system now, you're looking at a sagittal section through the male reproductive system and the male pelvic cavity. The way that we will work through this lecture is we'll begin with the scrotum, which is this structure here, and then we'll look inwards at the testes, and that's where sperm are produced. Sperm then make their way out of the testes. Oops, I'm going the wrong direction. Sperm make their way out of the testes into this structure here, this little comma-shaped structure called the epididymis. And those sperm gradually make their way down to the tail of the epididymis. And then upon ejaculation, those sperm leave the epididymis, make their way up this tube here, so they're traveling into the body now, over the bladder, behind the bladder, and it's kind of hard to track, you lose that structure here. But then sperm mix with some fluid from this structure called the prostate gland, and those sperm enter now the prostatic urethra, then enter the membranous urethra, and then we'll enter the spongy or penile urethra, and then finally make their way out of the body. So we're really going to follow where sperm begin their development, how they get from outside of the body into the body, and then outside of the body again. So it seems like kind of a redundant system. Why have the sperm made outside of the body only to go back in the body to get out of the body? And one of the reasons for that is that sperm don't have the ability to grow properly or develop properly at 98.6 degrees, which is body temperature. So we're not entirely sure why, but they need to develop at a little bit lower of a temperature, somewhere between 93 and 97 degrees Celsius. So the testes, which make sperm, the testes, which make sperm, are located outside of the body to help lower the temperature within the testes so that sperm development can occur properly. Okay, so let's have a look at the scrotum first. This is the sac of skin and superficial fascia that hangs outside the body, outside the abdominal pelvic cavity, at the root of the penis. And the scrotum is responsible for suspending the testes, the structures, the organs that actually create sperm. So in pink here, you see that this is the scrotum right here, and you can see the testes. The right side is the deep side, so here is the one of the testes, and then over on the left side, what you see is the testes surrounded by a muscle that we'll talk about in a second. So the left side here is the more superficial side. The testes are separated by a structure, so they're not one large sac. There are two separate sacs separated by what we call a scrotal septum. Sometimes that's called a midline raphe. It's just a connective tissue structure that separates the two into completely separate compartments. Go back to this picture right here. I guess they're not showing it very well, but in pink there, or in that peach color, that is the scrotal skin. And imagine little fibers, little smooth muscle fibers embedded in that scrotal skin. That would be the dartos. Go ahead and look at that cremaster muscle. The cremaster muscle is everything that you see here in red, that covering of the testes and parts of the spermatic cord. So you can pretty easily see those muscle fibers too. So all of that is the cremaster muscle. So that's going to elevate the testes and pull them closer to the body. The scrotum, remember, is suspending the testes outside of the body to maintain a lower, what we call intrascrotal temperature. So the scrotum would prefer, let me change colors here. The scrotum really only sees the temperature variation of somewhere between like 93.5 and 97 degrees Fahrenheit. So the fact that the testes are outside of the body automatically decreases the temperature of the testes, but there are other mechanisms that help to maintain the intrascrotal temperature within this range right here. And so one of them is a muscle called the dartos muscle, and that's embedded in the scrotal skin, so muscle. That dartos muscle, when the smooth muscle fibers contract, they pull the testes together. So the pulling of the testes together helps to increase the temperature of the testes. Imagine kind of what you do when you're cold. You put the parts of your body closer together rather than further apart. It helps to decrease the surface area to volume ratio and helps to keep structures warm or prevents you from losing heat quickly. So this causes a wrinkling of the testes. And then there's another muscle called the cremaster muscle, and this is actually skeletal muscle, but there's no voluntary control over it. What the cremaster muscle does is it elevates the testes up closer towards the body when the testes get cold, or the reverse is true. When the cremaster muscle relaxes, the testes fall away from the body and their temperature will drop. So, in addition to those two muscles, there's a vascular structure that's called the pampiniform plexus. Remember that a plexus is a network of blood vessels, and in this case, it's a network of veins. And these veins surround a structure called the spermatic cord. One of the parts of the spermatic cord is an artery called the testicular artery. And the testicular artery is sending warm core blood, 98.6 degree blood, to the testes. What the pampiniform plexus does, since it's made of veins, these veins wrap around that testicular artery and pull a little bit of the heat away from the blood in that testicular artery prior to that blood making its way to the testes. I'll show you what that looks like here in a second. And then also embedded in the scrotal skin, there are the salty or the exocrine sweat glands that cool the scrotum and testes when they're too warm. Okay, here we go. So we're about to move on to the testes, but before I do that, I want to point out the pampiniform plexus to you. So here's the pampiniform plexus. I'll circle that in green, and it refers to this network of veins that you see here. So it definitely takes on the appearance of a plexus, right? And then that's pampiniform plexus surrounds this red structure in the center. I won't draw all over it. I'll leave some so that you can still see it. But that's a branch of the testicular artery. And remember that that testicular artery is carrying 98.6 degree blood towards the testes. But the testes, remember, they would prefer to be somewhere, whoops, that's a bad color. They'd prefer to be somewhere between 93 and 97. So what will happen is, I'm running out of colors. What will happen is some of that heat that's within that testicular artery actually gets pulled away, and let's do this. Some of that heat gets pulled away and makes its way into the blood that is returning from the testes. And the reverse would be, or the same would be true over here. Here's another branch of that testicular artery carrying warm core blood. Some of that heat will be lost to the pampiniform plexus, or lost to the blood in that pampiniform plexus. And that blood then gets carried away from the testes, and therefore so does the excess heat gets carried away from the testes. Okay, we're starting again with a clean slate here. Let's recall that the scrotum is the sac of skin that encloses the testes. So the testes in this picture are here, this kind of grayish blue circular structure. And the main function of the testes is to produce sperm, gametes. And another function of the testes would be to secrete testosterone. A few slides back, we learned about some of the functions of testosterone. And those testes are suspended outside of the body cavity through a structure that we call the spermatic cord. So excuse the analogy, but it's kind of like the spermatic cord is the string and the testes are the ball of the yo-yo. And that's kind of a good way to envision how those testes are suspended very gently outside of the body by this spermatic cord and then enclosed in the scrotum. So the spermatic cord is made of a number of things. I'll go ahead and highlight all of the structures that make up the spermatic cord. One of those is a tube called the vas deferens, and that vas deferens is here, and it's this pink line that you see. Another structure that makes up the spermatic cord is the pampiniform plexus, that blue network of veins that you see there. The testicular artery is also a component of the spermatic cord. And I should probably draw an arrow to that because it's not labeled here for you. So that's a branch of the testicular artery. And then lastly, not shown here, but I suppose we could draw them in, would be some nerves and lymphatic vessels as well. You don't need to know the names of any of those, but there would be nerves supplying the testes and lymphatic vessels draining fluid from the testes. So earlier I mentioned that the spermatic cord suspends the testes outside of the body, kind of like a string on a yo-yo. But the testes don't start out in infants on the outside of the body. In fact, during fetal development, the testes develop inside the body and have to be pulled through the body wall. And the location where they are pulled through the body wall is actually a canal. And so this is called the inguinal canal. And I'll have to draw around the spermatic cord. But there's this little canal. This is this passageway between the abdominal pelvic cavity. So that would be inside, and the outside of the body, which is here. So this inguinal canal during fetal development is open and allows the testes to fall from the pelvic cavity to the outside, and then as the individual grows and develops, then ultimately what happens is the inguinal canal kind of seals up with connective tissue all around it. But that always remains a weak point in the body wall of a male, and the connective tissue fibers there can easily be torn, and when that happens, the spermatic cord can actually twist and cause a very painful condition that we call testicular torsion. But also think about what is immediately superior to that body wall, right above the pelvic region. Small intestines are right there. And so if the tear in the connective tissue of the inguinal canal is large enough, the small intestines can actually fall out of the body into the skin. And so that needs to be surgically corrected. And that's what we call an inguinal hernia. So within the testes, there's a system of coiled tubules that we call the seminiferous tubules. And it's in the epithelium of those tubules where sperm are produced in a process called spermatogenesis. So I'm going to jump to the next slide just really quick to show you what the seminiferous tubules look like, and then we'll come back and discuss the remaining. So we'll go back to the slide we were on previously, and we just saw what the seminiferous tubules look like. They're those coiled tubes. We saw them, how they're separated into lobules by areolar connective tissue. And within that areolar connective tissue are some specialized cells. So these are called cells of Leydig. Sometimes they're just called interstitial cells, but these are cells that are found within that areolar connective tissue between seminiferous tubules. And what they do is they're the cells that are responsible for secreting testosterone. If you look at the wall of the seminiferous tubules, remember we're looking at a hollow body tube, and all hollow body tubes are lined with an epithelium. So in a few minutes here, we'll look at that epithelium underneath the microscope, but the epithelium is going to be made of some specialized epithelial cells. Since the function of the seminiferous tubules is to create sperm, there must be some cells there that will become sperm, and those are called spermatogenic cells, sperm-making cells. And then surrounding those spermatogenic cells, and kind of making a nice snug location for those spermatogenic cells to develop sperm, are Sertoli cells. Sometimes these are called nurse cells, and their job is to nourish and protect the growing sperm. We call those growing sperm spermatocytes. That just means sperm cell. And so they're protecting and nourishing those spermatocytes as they complete the phases of meiosis. Of this information. So here's a picture from the Complete Anatomy app. This entire circular structure is there's one testy, and then these coiled tubes that you see in here, all of those tubes are the seminiferous tubules. And the seminiferous tubules are compartmentalized into what we call lobules. So there's one lobule, here's another lobule, here's another, and so on. And in between those lobules, where you find the seminiferous tubules, there are these areas of areolar connective tissue right here. Kind of almost looks like the kidney, doesn't it? Where you have those renal columns and the connective tissue in between. Obviously, there's no relationship here, but this is how this organ is put together. So, just as a reminder, let me switch my pen color here. You would find some Leydig cells in here, or in here, always in that connective tissue, right? And the sperm are actually being produced in the walls of these seminiferous tubules. It's actually a really cool process. Okay. All right, if we were to look at a cross-section through a seminiferous tubule, let's pretend for a second here is that cross-section through the seminiferous tubule, and here in the center, that would be the lumen of that tubule, and then and the wall would be made up of epithelial cells, really specialized epithelial cells that you see here. And so if we look at the drawing, let me get a better picture here. If you look at, or better color, if you look at the wall, here's the epithelium, right? So that's the epithelium of those seminiferous tubules. And there, you can see the two cell types here now. Let me see, I'll use yellow. And the nurse cells are these cells that you see in purple and I'm outlining in yellow, and their job, remember, is to nourish the developing spermatocytes, or kind of assist them through meiosis. And then here are the spermatogenic cells here. And we can follow just one through the process of meiosis. And we won't go through all the phases, but this cell right here has given rise to this cell, which then goes through some changes and then goes through a division, right? And then creates two, and then those two both split into twos themselves, so there are four more. Anyway, I guess my point is that it's really neat to see that sperm are essentially born by these cells down here, those spermatogenic cells, and the sperm mature in complete meiosis going in this direction towards the lumen. So, over on the left-hand side, you can actually see the cells. This cell right here is going through its late metaphase, early anaphase, right there. So these cells right here, they don't look a whole lot like sperm, but as they move towards the lumen, they mature even more, complete the process of meiosis, and will eventually even develop the beginnings of their tails, and also then become immature sperm, and their heads are kind of embedded in the wall of the seminiferous tubules for some time. So those were the cells that make up the epithelium, but I want to point out too, just like epithelial tissue anywhere else in the body, it's going to be anchored to some other tissue via a layer of connective tissue. So that's what you see there that I've highlighted in yellow. And that connective tissue creates what we call the blood-testis barrier, kind of similar to the blood-brain barrier, in that here we've got a blood vessel. Oh, that did not show up well. Here we've got a blood vessel, but it is physically separated by a decent layer of connective tissue right here, so that anything that might be traveling within the blood, like a pathogen or some harmful protein, it has a very difficult time getting through or diffusing through that thick layer of connective tissue. So it creates this nice barrier between anything in the blood vessel and the cells that are so important for reproduction. That's the blood testes barrier. So here's a picture right out of your textbook, and we lost a little bit of the formatting, but I think it'll be just fine. It shows you just in a different view what we were looking at. So here's the lumen, and so that's where the sperm will eventually end up. In the earlier picture, I showed you some sperm that had, that had their heads kind of embedded in the wall or in the epithelium. They'll eventually come loose and come out into the lumen of the seminiferous tubules. The picture here shows you the interstitial cells. Those would be those Leydig cells. Those are way up here. And the picture also shows you the spermatogenic cells here. This author's is using the term spermatocyte for those cells, and then giving you also other terms for the older versions. So as those sperm move towards the lumen, they're maturing, and then they do take on different names. Like this one right here is called a spermatid, and then, once it's got its tail, and its tail's hanging out in the lumen, technically it's called the spermatozoa. That's kind of beyond the scope of our course. You can just refer to all of these as spermatocytes, okay? So those are the spermatocytes that are undergoing meiosis. They're undergoing meiosis from the outside of the tube towards the lumen in that direction. And they'll eventually then pop off into the lumen. Nurse cell is pretty easy to see right here. And that's going to be one of those cells that's going to nurse or nourish and protect the spermatocytes as they go through meiosis. Here's what a sperm looks like, and it's kind of neat. It's a really interesting structure. You're not going to be tested on this, but I like to include it. And the way that I kind of think of sperm is that it's kind of like a rocket, and many rockets have some sort of payload on the front or something they're delivering. That's what rockets do, right? They deliver something somewhere at an extraordinary speed. So the thing that is being delivered is the DNA. Inside the nucleus of this part of the sperm is the DNA. That's what's meant to get to the ovum to help create that new human being. So that's the really important part. And that head of the sperm is just really a package of DNA. There isn't a whole lot else there. It's just really the package of DNA, and it's surrounded by, yes, of course, the plasma membrane, because this is one cell, right? This is not several cells. It's just a strangely shaped cell. So there's, on the outside, there's the plasma membrane, and then deep to that is what we call the acrosome. I like to think the plasma membrane and the acrosome like a helmet. And that plasma membrane and the acrosome are helping to protect the DNA here in the nucleus, right? We wouldn't want that nucleus to rupture prematurely and spill out the DNA. We only want that to happen when the sperm finally meets up with an egg or an ovum. And then another thing that happens is the acrosome right here. It's also called the acrosomal cap. That's got enzymes in it that start breaking down the plasma membrane of the egg as soon as it gets there, so that sperm can quickly deliver the DNA. So that section that you see that's circled here, that's just the head of the sperm. Okay, so that's just one part of the cell, but something has to be driving that head or driving that DNA towards its destination. And the structure that's doing that is the tail. It's a really highly modified flagellum. Well, in order for that tail to move back and forth and propel the head, where the DNA is, towards the egg, there have to be some engines or some structures that are able to make fuel for that tail to whip back and forth. And the structures that do that are the mitochondria, these green structures that you see here. They're modified mitochondria, and they're going to be constantly making ATP. So the tail, when it's time to fertilize an egg, that tail can whip back and forth to deliver the DNA. So the mitochondria and the tail here are like the rocket. And the rocket provides the energy or the fuel for delivery of the package, which is the DNA. So it's kind of a really cool setup that you see here. Oh, one other thing I want to mention is most living cells use glucose as a source of energy. You know that glucose goes into the cell. The cell carries out cellular respiration in the mitochondria, and out pops ATP. Well, human sperm don't use glucose as a source of energy to make ATP. They use fructose, which is very similar to glucose, but it's got its differences. And fructose can be used by the sperm to create ATP, just like glucose normally would. And so the sperm, we're not entirely sure, but we think that utilizing fructose as a source of energy reduces competition between sperm and any other organism that might be in the vagina when the sperm gets there. So if bacteria or other pathogens are in the way, or other commensal organisms are competing for glucose, well, if sperm uses fructose and not many other organisms do, then sperm have an advantage. There's an abundance of fructose for those sperm to use as a source of energy. So from the seminiferous tubules, the sperm will then be transported to a second structure called the epididymis. The epididymis, you may have seen it spelled like I have here with a D-Y, but you may also see it with a D-I-D or D-Y-D. Anyway, that the epididymis is a structure that lies not within, let's erase that, posterior to each testicle. And it's a coiled tube, just like we saw the coiled seminiferous tubules. And since it's a body tube, it's going to be lined by epithelial tissue. In this case, ciliated simple columnar epithelial tissue. So the sperm move from the seminiferous tubules into the epididymis. And what's happening in the epididymis, or the function of the epididymis, is the sperm will stay there for almost 3 weeks. And in that time, they're going to finish maturing. They're going to be transported from the head of the epididymis to the tail of the epididymis. And then once at the tail, the sperm will be stored there until ejaculation. So it's about a 20-day journey, 3-week journey from the head to the tail of the epididymis. And at the tail of the epididymis, there's a secretion of the epididymis. You don't need to know what it is, but it prevents the sperm from maturing any further. So once the sperm get to the tail of the epididymis, they're pretty much in their mature form, but still some changes need to be made. But what happens is we don't want that to occur too quickly, because the sperm are going to not have a whole lot of energy. They're going to use all that energy just to find the egg. So we don't want them to be utilizing a whole lot of energy and doing their cellular thing while they're waiting to be ejaculated. That time may be uncertain. So there's a secretion that kind of puts the sperm in a dopey state, kind of a suspended animation state. It just decreases some parts of their metabolism to just barely minimum to keep the cell alive. And that secretion then helps preserve sperm until the time that they're ejaculated. It doesn't allow them mature too quickly. When those sperm are ejaculated, there's smooth muscle in the tail of the epididymis that's going to force the sperm up and out of the epididymis into the next structure, which is called the vas deferens or the ductus deferens. Let's have a look at the epididymis, and I've labeled some things here for you already. So this region is the head of the epididymis right here, and then you have the body, which is this main part, and then you have the tail down here. So sperm are going to be, remember, they're going to be made inside these seminiferous tubules, right? And eventually those sperm get transferred to this network that you see here. And that's a structure called the rete testes. And the rete testes, this network of tubules, are going to transport the sperm to the head of the epididymis first. And from there, then the sperm will move slowly, right? This is gonna take it about 3 weeks. You're gonna have sperm move from the head down to the body, and ultimately they end up at the tail, where they're gonna be stored and prevented from maturing and swimming around too quickly. From there, the sperm are going to move up into the next structure that we're going to talk about here in just a second, and that is what we call the vas deferens or the ductus deferens. That's this word that you see right here. And that's this tube. It starts out coiled, and this is the tube that's going to carry the sperm from outside the body all the way inside to the body. Okay, so both the epididymis and the testes sit outside the body, but earlier in the lecture, I mentioned that they don't start there. During embryological development, the testes actually start out developing right next to the kidneys. So these are what will become the testes. They have a different name here. And you see how eventually they drop. So they move further away from the kidneys. And then they're going to drop down to the pelvic floor, closer towards that inguinal canal. So by about 4 months of embryological development, they're very close to that inguinal canal, where they're going to be pulled through the body wall and end up in the skin and superficial fascia of the newborn. So we can look just a little bit closer here. Now we're at 7 months of embryological development, and this is the what will in the adult be called the inguinal canal, and you can see the testes being pulled through there on either side. And then right about the time of birth, the testes will now almost fully be outside the body, suspended by the scrotum. And then eventually what should happen is the connective tissue here will kind of seal up around that spermatic cord, and then keep everything organized and straight. The spermatic cord, the testes, keep all of those things from getting all tangled up in the scrotum. So this is really where we ended up. We ended up learning about the epididymis right here. In this picture, you're looking at a posterior view of the urogenital system. And what we're going to talk about now is where the sperm go after they leave the epididymis. They make their way into this tube called the ductus deferens. So ductus deferens is also known as the vas deferens, and that's this tube that you see here. So the sperm, let me change my pen here. I'll have to use black, otherwise it won't show up. The sperm are going to, upon ejaculation, get pushed via peristalsis up this tube into the body, and then we kind of lose this, lose track here. I'll pick that up here in a second. Let me tell you what the ductus deferens looks like. So it's a tube lined with epithelial tissue. Specifically, this tube is lined with pseudostratified ciliated columnar epithelial tissue, and then outwards from there would be a layer of smooth muscle, and that smooth muscle is going to contract and via peristalsis transport sperm against gravity up into the body. So the function of that ductus deferens is to transfer sperm from the epididymis, which was down here, remember, right? To the base of what we call the seminal vesicle. And actually, we're going to just look at... So let me tell you what seminal fluid does. Seminal fluid kind of wakes up sperm, and remember earlier from that secretion in the epididymis, the sperm were kind of in a dopey state, not really able to do much except for exist. Now, once the sperm are mixed with seminal fluid, it wakes them up, and most of that seminal fluid, as I mentioned earlier, or a large amount of it, is fructose, that sugar that the sperm are going to use for energy to swim towards the egg. So that seminal fluid wakes the sperm up and provides them with fructose for their journey. Now, even though the sperm are awake, they're activated, they're not actively swimming yet. Their flagella aren't beating, they're not moving. Okay? That's more of a last minute thing. The sperm then are... Okay, so let's go back here real quick. Oh, no, just kidding. Next picture here real quick. I want to show you where the sperm meet up with the seminal vesicle. So I'll use, let's use green. So here is the vas deferens that you're seeing on the right. And sperm are going to be transported through that vas deferens and ultimately end up in this space right here, which is called the ejaculatory duct. And then over off to the right, you see this structure. That's a seminal gland. And that's a gland that has a whole lot of spaces or sinuses. So the product that this seminal gland is making gets secreted into those spaces and is stored there for use upon ejaculation. So upon ejaculation, when the sperm make their way into the ejaculatory duct, that seminal fluid will make its way also into the ejaculatory duct. So essentially, seminal fluid and sperm meet up and mix in the ejaculatory duct. From the ejaculatory duct, I'll just use a different color. The yellow color will represent sperm mixed with seminal fluid. So the sperm and the seminal fluid are going to make their way into this section of the urethra, and if you recall, that's the prostatic urethra, because the gland that you see right here, this donut-shaped gland, or actually it's almost like a heart-shaped gland, that's the prostate. And the prostate gland rather, then is going to mix its fluid with the sperm and the seminal fluid. The sperm and the seminal fluid is yellow, and let's just say that prostatic fluid is represented by this red color. So the prostate is going to secrete its secretions and mix that with seminal fluid and sperm. Go forward. So that's going to bring us to your slide so it's easier to see. What I'm going to do is go ahead and erase some of this stuff. I guess I don't really have to. So that seminal fluid plus sperm make its way into this section, which is going to be the membranous urethra. And this membranous urethra is actually embedded in what we call the urogenital diaphragm. That's just the skin, connective muscle, nervous tissue. That's just the tissue at the base of your torso. It's what's separating your insides from your outsides. That's the urogenital diaphragm. So that in that urogenital diaphragm, you see embedded are glands. They're called bulbourethral glands, but they can also be called Cowper's glands. Both are acceptable to, again, the prostate gland. I kind of said already most of this to you. But I didn't tell you what it was made of. It's made of a mix of connective tissue or fibrous connective tissue and muscle, smooth muscle, so that it can contract a little bit to squeeze that prostatic fluid into the prostatic urethra. Prostatic fluid is alkaline, meaning it has a high pH, somewhere over 7, and that's going to neutralize, the acidity of the vagina, and it's also going to activate sperm swimming or sperm motility. Let me elaborate on the alkalinity of the secretion. So the alkaline secretion of the prostate gland kind of coats the sperm in a protective alkaline coating, so that once they make their way into the vagina, this acidity of the mucus in the vagina doesn't break down the sperm before they can get up to the egg in the fallopian tubes. So that's alkaline secretion is just a coat of armor, really, so that the sperm can get to where they need to go. That on an terms. So from the prostatic urethra, sperm is going to get sent into the next section of the urethra called the membranous urethra. That word right there. And that's really just this section right here. Let's have a look. Okay, here we go. And those bulbourethral glands also secrete an alkaline mucus into the penile urethra, which is this whole section of urethra right here. Remember that in males, the penile urethra serves two functions. It's going to carry urine out of the bladder, but it's also going to be carrying sperm. So that, pardon me, that secretion of the bulbourethral glands comes out just prior to sperm and seminal fluid. And essentially what that fluid does is it rinses the penile urethra just seconds before the sperm come passing through, so that the penile urethra is rinsed of any pathogens, but also makes sure that the pH is correct for the sperm, so that alkaline mucus will increase the acidity of the urine that may have just recently passed through. And the last slide then is on an external structure called the penis. And this is the structure that is really meant to deliver sperm into the female reproductive tract, which is an internal structure, right? So there has to be something external that can be fit into something internal in order to get the sperm and the eggs together. We say that the penis is attached at the root, or the root of the penis is right here. You can kind of think of that as being the base. And then it has, the remainder of it is the shaft that ends in this bulbous structure right here called the glans penis. Glans, not Glands. DS. It's not a gland. It's not an exocrine gland or an endocrine gland. It's not a gland. It doesn't secrete anything. Okay, so be mindful of that spelling. The structure of the penis is such that it's made of what we call erectile tissue. And erectile tissue is essentially a whole bunch of connective tissue, so a spongy network of connective tissue that has many spaces in between. So if we look here in blue, these blue columns that you see here, the light blue would be the a little bit more densely packed connective tissue. And then what you see on the inside, all of these darker blue spots, those are spaces where blood can collect. So let me tell you what the names of those are, and then I'll tell you what these erectile tissues do. So there are 3 cylindrical columns of erectile tissue in the penis. There's one corpus spongiosum, and that would be, let me just change my pen so it's easy for you to see. The corpus spongiosum surrounds the urethra. So it's all of this, and then all of this on the other side. And of course that goes all the way around, right? You're just looking at it in a two dimensions right here. But it would completely encircle the urethra. Another way to look at it is, here we go. So there's the urethra, and here's the corpus spongiosum, so it completely encircles it. So there's 1 corpus spongiosum and 2 corpora cavernosa. Those are the other erectile tissues, and I'll outline those in yellow. So corpora cavernosa, they're actually located dorsally. Let's look at it in cross-section. Okay, so here would be corpora cavernosa, and so would there. So the function of having this erectile tissue is that upon the appropriate nervous stimulation, the blood flow to this area, to the erectile tissue, increases. So you see that blood vessel right there? So the blood vessels, and all around, all the capillary beds surrounding, blood rushes to that area, and that blood collects in those spaces of the erectile tissue. And that causes the penis to grow in length and width and becomes a hardened structure that then is an effective tool for delivering sperm inside the female body. So that is the end of the male reproductive system lecture. If you have any questions, please feel free to get in touch with me on Canvas or via email.